Hello, and welcome to what is now going to be our final video for Biology 1410, Introductory Biology. I'm only going to do one video on biotechnology because I know at this point you have to be tired of me at this point in the semester. So what I'm going to do, because biotechnology is really the application of what we have learned all semester in biology. And biotechnology is constantly changing. I'm going to have some other videos made by other people for you to watch because I want you to see some of the cutting edge stuff that's happening with DNA technology and things that can be done. Okay? So really when we're talking about biotechnology, for the most part, what we're talking about is manipulating DNA. So we're manipulating that genetic information in some way. And there are a lot of different ways how that DNA can be manipulated and how it can be used in day-to-day life. Okay? So with biotechnology, one of the applications is going to be genetic engineering. So with genetic engineering, we're talking about taking the genes from one organism and transferring it to another. And there are a lot of applications where that is something that you may come across in your day-to-day life. It used to be way, way long ago, back when I was a little kid, if you were diagnosed with diabetes, and we're talking type 1 diabetes here, where you need to have insulin injections, it used to be the source of insulin was actually from pig pancreas. So they would go to slaughterhouses, they would pull the pancreas out, they'd chop it up, they'd isolate the insulin. But the problem with that is there was a lot of variation in the amount of insulin they would get from the different tissues that they collected from. So what they're able to do now is they have taken the human gene for insulin, inserted it into a bacterial plasmid, so a short bit of bacterial DNA, and then they transfer that plasmid to a bacterial cell and essentially use the bacteria as a factory for making insulin. So in terms of genetic engineering, anytime you see a little R in front of something, okay, like insulin, although actually you'll see an H with there also. It's going to be the human recombinant protein, meaning that particular protein was made in a bacterial cell, and then the protein, in this case insulin, was separated from the bacteria. And that's now what most people inject if they are insulin dependent. It's a recombinant protein, and that's what it means for something to be recombinant. You have two different species involved, in this case human and bacteria, okay? With the bacteria essentially being exploited as factories for making protein, because the process of transcription and translation in bacteria is very similar to what we have in eukaryotic cells. There are some slight differences. Not all proteins can be grown in bacterial cells, but insulin is one example that can. Okay? So we've got genetic engineering as one of our applications of biology. Another one that has made some progress, although maybe not as much progress, is the process of gene therapy. Okay? Gene therapy is going to involve replacing, we'll call them defective, because right now gene therapy is only targeted at disease processes, all right? And that's less controversial than targeting gene therapy towards other processes. But for now, we're going to stick with disease processes. So we're talking about replacing defective genes. So people who have sickle cell anemia or cystic fibrosis, those are two examples of mutations that are good candidates for gene therapy. Because if we can replace their mutated copy with a good copy, their disease process ideally will resolve. It'll no longer be a disease process, all right? There are lots of technical issues with gene therapy though, okay? And hopefully I'll have some videos for you to watch on that, all right? So genetic engineering, gene therapy, and then the other thing that we're going to have is going to be DNA fingerprinting. Or forensics, okay? If you're like me and you like watching shows like Bones or CSI or shows along those lines where you're using genetic information to identify who committed the crime, that's what we're talking about with DNA fingerprinting, okay? Because everybody has a unique DNA fingerprint unless they happen to be an identical twin, okay? That unique DNA fingerprint can be used to identify who the criminal is. So if we're talking a murder case and somebody leaves DNA behind, that DNA left behind at the crime scene can be used to identify who the person was who committed the crime, okay? The other application for DNA fingerprinting is going to be determining who the parent is. And on this end, sorry guys, we're using DNA fingerprinting to find out who the dad is, because there's usually not any fuzziness on who the mom is, because the child is born from the mom, unless we're talking about use of surrogate eggs, okay? But I'm talking in terms of parentage. It's used to identify who the father is. Because in terms of DNA, all right, I've said this before, 50% of the DNA you get is from your mom, 50% is going to be from your dad, okay? That's going to make up your 100%. So in terms of using DNA fingerprinting to identify the child and who the child's father is, child has to have all of the genetic markers that are used they have to be present either in mom's sample or in dad's sample, okay? And I'm going to try and find some videos on that for you that'll explain things in a little more detail, all right? So that kind of wraps things up with biotechnology being the application of everything that you have learned this semester, okay? And how that biology can be used therapeutically, forensically, and other ways as well, all right? So thank you for your attention. Good luck with the final exam. If you have any questions, please let me know.